Hello, this is Catherine ASMR, and today I'm going to be taking you through the process of how a single cell becomes an entire human being, which is made of trillions and trillions of cells of all different types. So to do this, I have my trusty whiteboard just next to me here. I had to pre-prepare some diagrams because this would have just taken far too long to draw out, but I will trace over them purely for ASMR effect. So here we start at a single cell, and this is when the sperm first meets the egg and they form something called a zygote, which is essentially just the fertilized egg, but it's what happens when the two cells fuse together. And what's interesting is that those two cells just become this one cell here. And this one cell is completely undifferentiated, which means that it's not any specific type of cell, but it does have all of your DNA in it, so it has the potential to become any type of cell, because all of your genome, which is the total sum of all your DNA, is contained within every cell you have. And we'll explain how they become different cells and how they become different from one another later. So the first thing that happens is obviously a cell, and then I just want to make sure you can see, then it becomes something called a blastula. And so what this just means is that the single cell divides into lots and lots of cells. That's the first process that happens in the journey towards becoming an entire organism, in this case a human being. So what happens is that it starts to rapidly divide, again just the same type of cell at this point, and all of these cells are called blastomeres, and really this just means dividing cells in Greek or Latin, and it becomes the blastula, which is the collection of blastomeres. And at this point, this here is a blastocele, which is a cavity that begins to develop in the blastula, and actually it's the precursor for the first organ we have, and this organ becomes the gut. So before we have anything else, before we have a brain, before we have a heart, we have a gut. So from this point, the process of forming the gut, as well as some other things, is gastrulation. So I want to make sure that you can see this. The blastula becomes the gastrula. So you can see that there. I'm just going to put that away because you can't see it and me at the same time, but I'll describe what's visually going on. So what happens to the blastula is it goes through a process called gastrulation and turns into a gastrula. And at this point you've got your blastocele in the middle, and you've also got 3 layers of distinct cell types, so the cells are starting to differentiate slightly. So on the outside you've got the ectoderm, in the middle you've got the mesoderm, and around the developing gut, the digestive tube at this point, called the archenteron, you have the endoderm. So it goes archenteron, the cavity space in the middle that becomes your gut, endoderm, mesoderm and ectoderm on the outside. And then you have something called a blastopore at the bottom of the developing gut, and this will eventually develop to form your anus. Although interestingly, this is not the case in all animals, as in mollusks and I think it's worms, this becomes their mouth. And what's really interesting about these three distinct cell layers is that whether a cell is derived from the outer layer, middle layer or inner layer, it decides what kind of cells it has the potential to turn into and which organs can develop from that area. So around the digestive tube there, the endoderm, that can form the stomach, the pancreas, the intestines, all those organs sort of around there. The mesoderm, the one in the middle, can form the bones, muscles, circulatory system, the reproductive organs, and the outer layer of the embryo, the ectoderm, can produce the epidermis, which becomes the skin layer that we all have on our bodies, and it also goes to form our central nervous system. So this is where I will get the board back up to help me demonstrate this. So just to give you a little refresh, I'll show you the three layers again, and I'll pop out of the frame, I think. There we go. So we have the endoderm, mesoderm and ectoderm, which all turn into different things eventually as it becomes a more developed embryo. So one of these processes, which is very important, is a process called neurulation. And what happens is the mesoderm, the middle layer, it develops the notochord, which is the foundation of our nervous system essentially. And that notochord is an organizer, so it sends molecules up to the ectoderm, the outer layer, to begin developing the neural plate, which is here. And as neurulation takes place, this begins to happen where a tube begins to form, and if you can see there, that is the neural tube, and that becomes our spine. So that neural tube is obviously very important. It's the base of our spine and brain. It develops these things, and then the neural crest cells at the top in green there, there are neural crest cells that they are cells that migrate across the embryo and essentially just create the nerves of the embryo. So obviously we need nerves to do everything. We need nerves to move any muscle, anything like this, or feel pain or feel touch or anything like that. So nerves are very essential. What's interesting about neural crest cells is the fact that they are involved in some other processes as well. So for example, in dogs, something really interesting has happened since we've domesticated them, and dogs' ears in general have become floppier, and this is because neural crest cells are largely involved in ear formation, and as we've selected for tamer, more loyal dogs as we've bred them through the ages from wolves, we've unintentionally changed their neural crest cell function, or rather we've bred for a certain neural crest cell function that just happens to be linked with behaviour. And it has just led to them having floppy ears because their neural crest cells behave in a different way, which I find pretty cool. So that's the brain and spinal cord developing now. We're in that process. Another thing that happens is the development of the somites. And so, as I mentioned before, the mesoderm goes to create things like our circulatory system, our musculature, our skeleton, all of that stuff, our vertebral column, so basically just this area. And this is what an embryo kind of looks like for a portion of its life. So, you know, we've got essentially an overhyped ball of cells, and then we've had the central nervous system, brain and spine formation, or at least the beginning parts of it. And now we have a very important structural step, which is the development of the somites here. And I don't know if it's very obvious there, but it's basically a baby 8-pack, but I think there's many more than that. This is the developing brain here, and the somites, these little muscles here, this is where the vertebral column extends from, and the bones, your ribs, etc. So it all grows from that point. Now at this point I want to get a little bit more technical, because I've explained what's happening but not why it's happening, how it's happening. So the way that all of these cells become differentiated, because obviously you can't have one cell performing absolutely every function you need. A neural crest cell is going to have a specific function. The eventual production of neurons, brain cells, is going to have a very specific function. So how does that all happen? It's all about the way that genes are expressed. Genes are either switched on or switched off in different stages of development. So the undifferentiated cell at the beginning, basically none of the genes are off and it can turn into any cell. But as you continue through development, different genes are switched on. And this is done with transcription factors, which basically allow proteins to be transcribed from your genetic code, your DNA. So the presence of different transcription factors allow certain genes to be decoded and a protein to be made. So that is how different cells have different functions, because essentially the principal function of DNA is to create proteins. Any process in your body is taking place because of proteins, and obviously, especially in today's protein obsessed age, we really think of protein to being very important for muscle, but different proteins aren't just about that. They're for all sorts of functions. Any enzymes in your body, cell wall components in your cell wall, just everything basically. And you need different proteins to do different jobs. For example, for your immune system, those cells are all different proteins that do a specific job of, you know, taking down pathogens and whatnot. So yeah, DNA is transcribed to produce the specific proteins that make a cell a specific cell. To make a brain cell, you obviously need the right proteins to allow it to perform that job. In a muscle cell, you need the proteins to do that job. They're obviously not doing the same things. Your muscles need to contract, whereas your brain cells need to send signals, so it's going to involve very different proteins. What these transcription factors do in these stages of development, so that different cells can specialize and do different jobs, is they turn genes on and other genes are inactive. And then these cells, they collect into collections of cells called tissues, which are all the same type of cell, but there's lots of them, which allow it to perform a function at a greater capacity. And then you have organs, which are comprised of different tissues containing the same types of cells, but you've got different tissues all contributing to the functioning of that organ. So that's how that works essentially. So for example, in a muscle cell, a transcription factor will tell a muscle cell to become a muscle cell, and it does this by activating the right genes for the muscle cell. For example, there is a protein called myosin which enables your muscles to contract, and obviously that's very specific to a muscle cell, and you have stuff like that happening all over the body. The gene switching on thing is just about making sure that each cell has the right proteins, and that will define the structure and function of the cell. Also, early in the development of an embryo, when it's kind of being decided, you know, where our head goes, where our arms go, where our legs go, as well as where all our tissues and organs go. You don't just get a person with a stomach up here and a heart down there. It's all pretty much according to the same body plan. These are controlled by homeotic genes, which essentially does just that. It decides where our bits and pieces go and where our tissues and organs go, and they decide, you know, how they're spatially separated, as well as where they are. And this is through the presence of transcription factors called morphogens, and they switch on these genes at that stage in development. Overexpression of a homeotic gene can cause crazy things to happen, not so much in humans because there's so much regulation that it would be rare, but in cases where it doesn't make a huge amount of difference, mutations can still happen due to overexpression of a particular gene. For example, there are plenty of people out there with 6 toes, and this is as a result of overexpression of the homeotic genes. This is called polysyndactyly. But a really interesting and simple example of how homeotic genes kind of work and are affected by morphogens is in an insect. So in an insect you have a defined body plan, not all of them obviously, but in most insects you have a defined body plan of a head, a thorax and an abdomen at the back. So in this case it's found that their genes, these homeotic genes, are very highly expressed in the head. The same genes are expressed intermediately in the middle, so slightly less expression, but still exactly the same genes are being expressed, but there are less morphogens, and so there's less expression of these genes. And in the posterior, in the abdomen, there's the least expression of these genes, so there are fewer morphogens influencing that, and then that is how it knows to turn into the abdomen, and then all the cells that need to be in the abdomen and all the cells that need to be in the thorax, all the cells that need to be in the head, that determines that they're going to be there as well. This is called like determining the axis of a body, and obviously it happens in embryos, human embryos also. Of course there are many more processes that go into making an entire human being or an entire organism of any sort, but I just thought I'd go through the main mechanisms and sort of the main structures and where they come from and why they develop as they do. I hope I've done a reasonable job considering it's such a complex topic. I know that I won't have done a perfect job. Obviously these aren't all the mechanisms by which just a single cell becomes an entire human being. There's obviously just a huge amount of complexity. You could go so deep down into it, but I just thought I'd share a few of the main mechanisms to help you sleep, or maybe just share a few interesting facts. And obviously I could give the muscle example about all of the different types of cells and tissues and organs in the body, but that would be an extremely long video. In fact, I would probably be dead before I managed to say them all. So just know that you can apply what was mentioned in the muscle cell to all the other cells. Essentially it all happens in the same way. There are so many other little processes that contribute to our formation. For example, apoptosis is a really important one, and apoptosis just means programmed cell death. And you wouldn't think that this is important in your development, but if we didn't have apoptosis, our hands would be like this. So before we fully developed, our hands pretty much are like this without any space between. And then as we develop into a fully grown embryo, we start to have programmed cell death coming down here, so that you can get these complex structures, and this is how we have our dexterity. And some people do still have webbing here, and it's not anything weird. We all have it at some point, it's just that most of us get rid of it in the womb. So yeah, I hope you've enjoyed, and I'll see you